Okay, let's go. We've lost 2 minutes. My God, we gotta catch up. So we left off talking about microbial nutrition, so how microbes are importing things like sugars or amino acids. So we left off talking about the simple transporters, and now we're moving on to the ABC transporters. And again, this should have some familiarity to you, because when we talked about the ABC or type 1 protein secretion system, this is actually highly related to that, except now the movement is in the opposite direction. So material is being brought in by these ABC transporters, and again, just like with the type 1 or ABC protein export system, ABC stands for ATP binding cassette. But again, there are 3 components to this. The energy source is inside the cell on the cytoplasmic side. It's the hydrolysis of the phosphoanhydride bond of ATP, generating ADP. That's what facilitates the import of a molecule here shown as this red dot. The neat thing about this system is that it has a partner protein in the periplasm, if we're talking about a gram-negative bacterium, or actually in the extracellular environment if we're talking about staphylococcus, another gram-positive. The solute binding protein, which has an incredibly high affinity for some type of a molecule, sugar or amino acid, perhaps it's an ion or something, so that it can bind it when it's at very, very low concentrations. So this is a very efficient way that bacteria can scavenge nutrients when they exist at low levels. So these solute binding proteins will bind to a solute molecule. The best studied of this is maltose in E. coli. Binds that disaccharide, delivers it to this component, which then will cause ATP to be hydrolyzed on the cytoplasmic side, and that molecule, maltose perhaps, is transported across. But it's important to note, because one of the next pathways that we talk about is going to modify the sugar in the course of bringing it in. This molecule being transported by the ABC mechanism is not chemically modified. It's the same on the inside as the outside. That will become, the significance of that will become more apparent when we talk about group translocation. Now, just as a means to show you why we care about this at all, other than people who are just interested in microbial nutrition, as I'm sure all of you are, just stay up nights thinking about microbial nutrition. But a lot of these pathways that we're talking about have actually been modified by bacteria, and they're a major reason for the drug resistance that we deal with today in terms of antibiotic resistance. So if you look at one of the mechanisms, so we talked about penicillin resistance and we talked about vancomycin resistance, and those were kind of enzymatically driven. So in the case of penicillin, you had the beta-lactamase, which broke down beta-lactams. So that's one way in which bacteria can become resistant to antibiotics. But a second major way that bacteria have acquired the resistance to antibiotics is to co-opt some of these pathways that we've just been talking about for either protein export or nutrient import, and just mutate them so that now, instead of transporting out what they normally do, they're transporting out things like amino acids, aminoglycosides. These are things like kanamycin, tobramycin you may have heard of. But what is happening here is that this bacterium acquired a mutation, or perhaps by horizontal gene transfer, it has acquired a whole new transport molecule that can now use a gradient of sodium to pump out antibiotics as fast as they come in. So what this does is this reduces the concentration of the antibiotics in the cytoplasm, so they can't have an antibacterial effect. So the bacteria just keep pumping these things out. And virtually every one of these systems has as its evolutionary precursor something that probably didn't transport antibiotics. some mutation occurred, and then by natural selection, staphylococci, which have these QACA and QACC transporters, have become very adept at transporting out of the cytoplasm some of these antimicrobials. Here is that system that we talked about in the ABC type 1 protein secretion system. Exactly the same. Here's that TOL-C protein that we talked about as a multifunctional protein transporter. Well, now it can hook up with this ACRA, ACRB complex instead of a protein that's being put out. Instead, what's being put out are a whole class of antibiotics. So this is part of the reason why it's important that we understand how these pathways work, because they're so critical to understanding how bacteria evolve to be antibiotic resistant. And that ABC family that I just talked about, here's an adaptation of that will also hydrolyze ATP, just like in the previous slide where I was talking about nutrient import. But in this case, the direction has been switched. So instead of bringing something in, it's pumping out any one of multiple drugs. So this is a very profound way in which bacteria have evolved the ability to become resistant to antibiotics. So now you realize why microbial nutrition is so important. So let's finish up with, well, we're not going to finish up with microbial nutrition just yet, sadly. Let's talk about this last active transport system. This is a very important system by a large number of bacteria. The system kind of varies from bacteria to bacteria, but they're all collectively referred to as group translocation. We're going to go through this system as it's best studied in Salmonella and E. coli and their relevance. But this phosphotransferase system, which is just another term for group translocation, is very widespread among bacteria. So it's a major way that bacteria can transport in sugars. In E. coli, the system brings in mannitol, it brings in glucose, it brings in mannose. It also can transport in fructose. These are just 4 of E. coli's most delicious proteins that it finds most nutritious. But this system is going to transport the sugar in, and it's going to phosphorylate it. So this is the case where the molecule being transported in becomes chemically modified. Each of the other active transport pathways that we've just talked about brings in a molecule without chemical modification. Requires energy, but that energy doesn't modify it. So it's an important distinction between this and the other pathways, is the chemical modification of the transported product. So let's start with this image. And so a nice thing about this image is pay attention to the colors a little bit, because the first 2 steps in this process are in green. And then the last steps are kind of in some shade of yellow. This can really be broken down into halves. The half shown here in various shades of green is a part of the pathway that's shared among all of the sugar transports. So this enzyme 1, or E1, and this HPR, or histidine protein, are going to drive the transport of mannitol, of glucose, of mannose. But those ones shown in yellow are now sugar specific. So there is a specific enzyme complex in the membrane of E. coli, Salmonella, Klebsiella, which will transport mannitol and nothing else. But it gets its energy from the first half of the system. The same thing with the glucose transporter of E. coli or Salmonella. It has a collection of proteins in its membrane that will transport glucose only. So these are individual. They all share this enzyme 1 and enzyme 2. So the way this system works, this group translocation system works, is that you have a phosphoryl or a phosphate cascade going on. Where, remember phosphoenolpyruvate from the days when you had to memorize the glycolytic pathway? You don't have to memorize the glycolytic pathway in this class, just so you can relax. But remember that phosphoenolpyruvate, that high energy intermediate near the end of the glycolytic pathway, is high energy. Here, the phosphate group, that high energy phosphate group, is taken from phosphoenolpyruvate and put on to enzyme E1. So shown here by this little golden sphere. This is the phosphate group. That phosphate group is then transferred to this HPR. H stands for histidine. So here's another example, kind of like the key A, the histidine kinase that we talked about for chemotaxis. Here's another example of how bacteria do phosphorylation somewhat differently than eukaryotic cells. Histidine phosphorylation in your cells and my cells is quite unusual, if it occurs at all. I don't even know if it occurs at all. But this is a histidine-rich protein that becomes phosphorylated. Now, at this point, this phosphorylated histidine protein can donate this phosphate group to any of the members of these different sugar import pathways, just depending on what sugar is available on the outside of the cell. E. coli, salmonella, whatever, loves glucose best. And we'll get into the mechanism behind this, but if you put E. coli into an environment where it has equal amounts of mannitol, glucose, mannose, or even if it has a huge excess of mannitol or mannose, but it has some glucose, this HPR phosphate is always going to put its phosphate group onto enzyme 2A, specific for glucose transport. E. coli will always use glucose in preference to any other carbon source because it's the most efficient. Other sugars like mannitol, fructose, mannose can be used. They're perfectly delicious, but they require additional enzymes to process them to put them into the glycolytic pathway. Even though you don't still remember all of the steps of the glycolytic pathway, you certainly remember the first molecule going into the glycolytic pathway is glucose. So if a bacterium like E. coli is transporting in mannitol or mannose, it has to then enzymatically break that molecule down into something like glucose or something else that fits into the glycolytic pathway. That's an additional use of energy. So the bacteria have evolved this really elaborate way, and we'll talk about the mechanisms later, whereby they will selectively bring in glucose until it's completely scrubbed from the environment, until it's completely gone. Then this HPR phosphate will then, if mannitol is present, will alternatively phosphorylate E2A of the mannitol import pathway. That phosphate group will be transferred to enzyme 2B of the mannitol phosphate. And now as mannitol is brought in, that phosphate is put onto mannitol. So here's that chemical modification that happens every time. So what's being on the outside of the cell is mannitol. What's actually imported is mannitol 6-phosphate. Same thing with glucose. Glucose, when it's being transported, that same phosphoryl group from HPR went to glucose 2A, glucose 2B, and then as glucose comes across this transmembrane protein, glucose 2C, that phosphate group is transferred to glucose, and so you have glucose 6-phosphate. In a way, this is kind of cool because, again, not remembering the glycolytic pathway, I'll just tell you that the first step of the glycolytic pathway that you all learned, the Embden-Meyerhof pathway, is glucose under the influence of glucose kinase makes glucose 6-phosphate. So this molecule, because it's now glucose 6-phosphate, just fits into the glycolytic pathway one step down. This kind of recoups the energy that was lost because a glycolytic pathway intermediate phosphoenolpyruvate was used up in the process. But now what fits into the glycolytic pathway is actually one step down, glucose 6-phosphate. But glucose is always brought in preferentially first. Now, in terms of the growth of bacteria and nutrients, typically, we don't use this kind of media very frequently in the laboratory because most bacteria, unlike Bacillus megaterium, which we occasionally use in the laboratory, don't have a defined media. So I put this example up of a defined media for Bacillus, and we're going to use a defined media next week in the laboratory, a different one, because it kind of illustrates, what a pain in the ass it would be to make up large amounts of a chemically defined media. Because you would have to weigh out specific amounts of dibasic phosphate, magnesium sulfate, all of these kind of things to give you carbon sources, phosphate, potassium, nitrogen, sulfur, magnesium, iron, manganese. All of the necessary nutrients, the micronutrients of the bacterium, could be made up this way, but it is a major pain. Instead, for the most part, whenever you have grown bacteria in the laboratory or will ever grow for the most of your career, you'll use what's known as a complex media. And after this next week in the laboratory, we will never use defined media again, mostly because it's me that has to make it, and it is a pain to make it. Instead, we use these complex media. So things that those of you who probably used LB, lysogeny broth, or sometimes referred to as Luria bertani broth, is a complex media. It has yeast extract in it, which is just autolyzed yeast. Beef extract, which my favorite, because this is what's scraped up off the floor of the slaughterhouse that we won't even feed to our cats and dogs. It's then enzymatically hydrolyzed, and we feed it to our bacteria. Now, the reason that this is undefined is because no one knows exactly what the concentration of any of the amino acids, vitamins, or whatever are in this media. And each lot is going to be slightly different depending on, did they scrape up more intestinal contents from the floor of the abattoir that day or not? Peptone is a peptic digest of meat. And again, these are great sources of energy and carbon for the bacteria. But in terms of nutrition, in terms of actually doing studies where you need to control the concentration, this is really not very good. This is actually what we would probably refer to as witch's brew. And so, here it is, eye of newt and toe of frog, wool of bat and tongue of dog, adder's fork and blind worm's sting, lizard's leg and howlet's wing, for a charm of powerful trouble, like a hell broth, boil and bubble. See, you get some literature when you come to this class, too. We're not done. Any Kipling fans? No? So here's from Kipling. Gold is for the mistress, silver for the maid, copper for the craftsman cunning at his trade. Good, said the baron sitting in his hall, but iron, cold iron, is master of them all. So what the hell does this have to do with anything? So obviously, because now what I want to talk about is one of the most important micronutrients that microbes need, and you and I need for that matter, and that's iron. And iron is a particular problem for bacteria, whether they live in the environment or whether they live in association with a vertebrate host, like pathogens that most of us are interested in. Because in the environment, because we live in an oxidizing environment for the most part, and you know that iron can exist in both ferrous and ferric, remembering back to Gen Chem, most iron in the environment is in its most insoluble form. So oxidized iron or ferric iron isn't very soluble in an aqueous environment. So it's difficult to come by for bacteria. Same thing in association with the host. Part of our defense systems against infection is built around the fact that we try to restrict iron from bacteria living in association with us. Because if a pathogen gained access to us and also had a very rich source of iron, it would have a very strong advantage. So you and I and other vertebrates have a very elaborate iron sequestration mechanism as part of our innate defense system. So things like ferritin, which is found within virtually all living cells as an intracellular store of iron. Transferrin, which is the iron-binding protein in our serum that tries to keep iron away from pathogenic bacteria, but still deliver iron to our cells. And the one I think is the most interesting is lactoferrin. So lactoferrin is an enzyme, a glycoprotein that we produce in abundance in saliva, in tears, but most especially in mother's milk. This is one of the major ways that mothers protect their infants against bacterial infection, because mother's milk is rich in lactoferrin, especially what's referred to as the colostrum, or the first milk that a mother, and this works for both humans and animals. The first milk given by a mother is very rich in antibodies and a lot of lactoferrin. What that does is it sets up a system inside of that baby or that small animal where iron is very restricted. So in the absence of a very strong immune system in a neonate, one of the ways that mother protects baby is to restrict iron. So it's a nice, you think about the beauty of mother breastfeeding her child. It's also helping to give immunity, both specific and non-specific. So basically, this is one of our major strategies for helping to deprive pathogens of iron that they need for cofactors, cytochromes, the same things that we use iron for electron transport to do respiration. Now, bacteria don't just take this lying down. They take this as a challenge. And so what bacteria have done, both environmental bacteria and pathogenic bacteria, is they've evolved the ability to secrete into the environment high affinity iron binding molecules known as siderophores. I pronounce them siderophores. I've heard a lot of people call siderophores. I think that's more the British and Canadian, I don't know. But you can pronounce it either way. But these are molecules that are secreted into the environment to bind iron, so that hopefully the microbe can now take that back up, because iron is so critical. So E. coli secretes one, and it's not the only siderophore that it secretes, but its best studied siderophore is called enterobactin. That's what's shown here. So enterobactin is actually secreted into the environment as one of these three catechol molecules. So it's unfortunately kind of shown here in its complexed form, where three different catechol subunits of enterobactin have come together, nucleating around an iron molecule, a relatively insoluble iron molecule. What this does, the collective action of all three of these enterobactin molecules binding to this, is it increases the solubility of that molecule. So now the molecule can be transported in water much more easily. Other microbes in the environment, especially in the ocean, because in many parts of the ocean, iron is tremendously limiting. So it's one of the major micronutrients that limits microbial growth in the oceans. A lot of microbes will produce this aquaquelin, which has a fatty acid tail. And so what happens is that these things collect into basically spheres or micelles, because it has this hydrophobic tail. But then sticking out from that sphere are going to be all of these molecules which have the ability to bind multiple iron molecules. So think of this micelle or sphere as just kind of randomly going through the ocean, collecting iron as frequently as it can. Now, none of these systems have a way to directly find their way back to the bacterium that secreted them. As a matter of fact, the chances of the bacterium that secreted this aquaquelin, or the E. coli that made either this enterobactin or hydroxynate, the chances that the bacterium that synthesized that ever sees it again, virtually nil. But it's a matter of collective activity. So if all of the bacteria in an environment are producing aquaquelin or enterobactin, some of their brethren may come in contact with aquaquelin and be able to bring it back in, or come into contact with enterobactin and bring it back in. But it's a completely random, diffusion-based phenomenon. So here's the mechanism, because this is what we understand the best, obviously, is E. coli. E. coli synthesizes and secretes that enteroquelin into the environment. This cartoon shows it being secreted as all three subunits together. I don't think that's true, but for illustrative purposes, I think this works fine. It can take relatively insoluble ferric iron, solubilize it, and it can now be transported across an outer membrane protein, a porin protein, that's specific. The only thing that this porin protein will allow to come across is going to be the enteroquelin iron complex. Nothing else gets through. There's no energy involved in this. This thing just happens to fit and get through. Other small molecules don't. Now, once that's into the cytoplasm, it's bound by, and we just talked about this type of an import pathway, the ABC pathway. So here's an example of one of those periplasmic binding proteins that I referred to generically in a couple of slides earlier. Sept, this protein, has the ability to bind to very small concentrations. So almost as soon as this enters here, Sept is going to bind to it. And then only in that bound form of Sept with the enteroquelin iron will it interact with its cognate or partner ABC pathway, Sept and Sept on the cytoplasmic side. I'm not really, honestly, interested in you remembering the names of these proteins. It's the principle that I'm interested in. Don't think that you have to memorize Sept, Sept. That's not important. It's the concept here. So this ABC pathway will now, just as we described a couple slides earlier, hydrolyze ATP on the cytoplasmic side, and the enterochelan iron complex is brought inside of the cell, where the iron will be reduced. Now, how is the iron reduced? If you think about the difference between the outside of the cell, here the peroplasm or the outside, versus the cytoplasm is a very oxidizing environment. So we're doing the cells and our cells are doing lots of oxidations of glucose and whatever we're using to generate energy. And when you do oxidations, you're taking electrons off things, right, by definition. So there are lots of electron carriers in the cytoplasm of the cell that are just looking for some place to dump their electrons, because electrons can't exist free in the environment. They have to go someplace. So these electron carriers are either looking to put their electrons onto the electron transport chain to generate proton motor force, or they can instead put their electrons onto iron 3 to generate iron 2. This enterochelan is so highly adapted to iron, it only binds the oxidized form. The reduced form is now freed into the cytoplasm to be used to generate hemes, cytochromes, all the things, for the most part, that you and I would use iron for. And now, this enterochelan molecule will actually dissociate into its 3 subunit parts and be recycled back out to go out and search for more iron in the environment. So it's a really elaborate way to bring iron in, and it kind of illustrates how important iron is for these cells. And I think I kind of got ahead of myself because, yeah, I mentioned that the cytoplasm is, I should have said, it's a reducing environment because there's lots of electrons looking for a place to go. So iron is, from the ferric form, is reduced to the ferrous form, and then the siderophore is recycled to pick up more. Okay, so I hope I remembered to leave this out of your PowerPoints, because this is just something I want to talk about in class, but it's not something that will be covered on exams. But this relates to global climate change, and so as citizens of the world, I think it's important that you understand that microbiology really needs to be understood as you begin to weigh in on policies that relate to global climate change. So I mentioned that iron is a limiting nutrient in the ocean. So where do you think the greatest source of iron in the ocean comes from? Anybody? I think I heard somebody say the desert. No, I didn't. I'm not one who can sit here and wait 8 seconds like I'm supposed to for rhetorical questions. Here's a picture from a satellite. And so here's Spain, the Iberian Peninsula, here's the Sahara. And so what you see is that while you and I right now are more worried about hurricanes coming across the Atlantic Ocean, you know, like Irma and Maria and all these terrible hurricanes we're having, but also those same winds that create or that help create hurricanes also blow an awful lot of sand from the deserts. And this ends up getting deposited in the water. So this is one of the major sources of iron in the water of the world. So if you look at, so here's a map that looks at air particulates. And so this is mostly dust in the air. And you can see as the colors become warmer, you have a lot more of soil particles largely in the air. So adjacent to very warm areas, so large, this is how iron is being deposited in the water. Now, if you look at oceans in terms of nitrate, so again, nitrate is another one, like iron, of a very limiting micronutrient in the environment. If you look here at nitrate concentrations in the oceans across the world in micromolar, you find that most of the temperate oceans tend to be very, very low for nitrate. So part of what limits photosynthesis and primary production in the oceans is the relatively low concentration of nitrate. But look at where nitrate is really high, down in the southern oceans. So, but southern oceans are actually not that rich in primary production. So that lack of primary production down here is largely due to the absence of iron. So if iron were added to these southern oceans, where there's lots and lots of nitrate, you could imagine that in the presence of sunlight, which there obviously is still in the southern oceans, there would be a lot more photosynthesis. So this has led people to propose an active measure to hopefully mitigate global climate change. So I think all of us know, as we think about how we're trying to mitigate global climate change, we think about reducing greenhouse gases like CO2 and methane and nitrous oxide. And that's a very good thing to do. But there are a large number of people who feel that even if we were able to get down to much lower levels of emission of these things, we may have already passed a tipping point. There may already be enough CO2, methane in the environment that even if we stop putting it in, we're going to continue to warm. And we'll talk in a later part of the class about melting permafrost and the methane that's coming from that and that feedback loop. But what your generation is probably going to have to deal with, or may have to deal with, if those people are correct, if we've passed a tipping point where simply stopping or reducing our greenhouse gas emissions is not enough, what's an active measure that we can take? So one of the things that's been proposed, and you may have heard of this before, is iron fertilization of the ocean. So people have done these experiments accidentally before. When a volcano erupted in Antarctica years ago, there was a big ash plume that ended up in the ocean, and there was consequently a huge algal bloom after that. But the idea being is that what people have proposed is to simply dump iron filings from ships into the southern oceans to increase the iron concentration, so that photosynthesis would now be increased by the phytoplankton there, which would suck CO2 out of the atmosphere. So can we actively begin to remove CO2 from the environment as opposed to just slowing our production of it? Could this decrease global warming? So once those phytoplankton die, so the attractive portion of this proposal is because those waters are so cold and so deep, that as those phytoplankton die, they will sink out of those surface waters, taking that carbon with them down to waters in the bottom of the ocean that are uniformly at about 3 to 4 degrees centigrade, where they will just rather lie there, becoming sediment and sequestering that carbon, taking it out of the atmosphere for centuries. So more phytoplankton, this is also part of the attractiveness of this proposal, is that more phytoplankton means more food for other species, so increasing the fisheries. So another problem that we obviously have in this world is our increasing human population, and how do we feed people? And so people have proposed that this could actually increase the fisheries to help feed the people. Now, of course, there are arguments against this as well. The greatest of which is obviously anytime humans ever think they understand how the earth works well enough that they can do something about it, they screw it up big time. So hubris is a big problem for humans. So these are things that you're going to need to think about, and you're going to have to be able to address. And so adding iron completely changes that biological community. So if you increase phytoplankton, yes, maybe you increase the fisheries, but think about that benthic ecosystem that's now being covered up with all of this dead algae. How is that being changed? That's not how that system has evolved to be. I don't know why my remote is not working. It will also affect the oxygen level. So because as that phytoplankton decays, oxygen is taken from the water. So we may actually end up with areas of reduced oxygen. And also, that microbial activity that's associated with reducing the oxygen can actually produce methane, which is many times more potent because of its molecular structure, many times more potent than CO2. So even producing smaller amounts of methane can have huge amounts. Nitrous oxide, even more strong a gas, or as a greenhouse gas. So to have any way to benefit a store of carbon, iron fertilization needs to be done in those southern oceans because there's so much nitrate there. So it's one of the only high nitrogen, low chlorophyll regions. That's what HNLC stands for, where the waters sink deep enough to take the ocean with us. So the only place that you can do this is in the southern oceans. But here's the weird thing. Look at where CO2 is. So here, the warmer colors on this map indicate higher concentrations of CO2. Not surprisingly, it's over the US and the northern hemisphere through Asia and Europe. It doesn't get into the southern. So the equator seems to kind of separate our atmosphere. This was something I never knew until I saw a map like this. So there's not much CO2 around these southern oceans to be sucked out. It's up here where we would need to get rid of the CO2 levels. So this is another reason why perhaps this proposal may not be the best way to stop global climate change. Okay, so back to reality, back to materials that I can ask questions on. Because now what we want to talk about is bacterial metabolism. So we've gone through the point where we talked about nutrient uptake and iron uptake. So now what is it that the bacteria do with these nutrients and iron? What's their metabolism? So that's the next section that we want to talk about. And so we've kind of touched on this a little bit already. But there are three forms of energy utilized by prokaryotes. And at least two of them are also used by eukaryotes. So phototrophy, using sunlight. Chemoorganotrophy, which is what you and I are doing as we speak, where we're using reduced organic compounds and oxidizing them to generate energy. And then the chemolithotrophy, this is the part that's uniquely prokaryotic. This is an energy type that eukaryotes have never been able to do, where you're using reduced inorganic compounds, oxidizing them to produce chemical energy to do work. So just some definitions. We don't really need to go through these very extensively. I just have this in there so that as we talk later, in other slides, you can look back for definitions, because you all probably remember these, but if you don't, they're here for you to review. So oxidation we talk about a lot, and remember that at the chemical level, that simply means the removal of electrons. So as we're oxidizing glucose, we're removing electrons from it. And reduction is the opposite, the addition of electrons. And in biological systems, you can usually recognize highly reduced molecules because the electrons are countered by having protons associated with them. So you'll see oftentimes the H associated with it. That means that that molecule is reduced. We're going to talk a lot about fermentation and respiration. So fermentation is something that bacteria are absolutely, you and I can do it a little bit. That's how we produce lactic acid. So if you know that side stitch that you can get from temporarily using up or generating lactic acid. But bacteria are kings, queens, princes, whatever, gender neutral you want to use, but they are wonderful at doing fermentations, various amounts of fermentations. Basically what these are oxidation reduction reactions where the final electron is put onto an internal electron acceptor, so something like pyruvate, as opposed to, this is more easily understood in terms of thinking of fermentation as the opposite of respiration. Because in respiration, which what you and I are doing as we speak, is we're taking those electrons and putting them on an external electron acceptor. So we breathe in oxygen. Oxygen is coming from outside. This is where those electrons will finally end, reducing oxygen to water. Bacteria do respiration as well. They also use oxygen, but unlike you and I, when they run out of oxygen, you and I would die. But many bacteria can switch to using nitrate, sulfate, sometimes elemental sulfur, all kinds of manner of things as a terminal electron acceptor. But those electron acceptors are always external to the material being oxidized. In fermentation, that same electron that needs to be dumped someplace, because electrons, they have to go someplace, goes onto a part of the molecule that's being oxidized. Remember that at the end of the glycolytic pathway, pyruvate can be reduced to lactic acid. It can have electrons added to that to generate lactic acid or lactate. That's a fermentation reaction, and it's not energetically as efficient as respiration. Now, two main means of energy conservation in bacteria, either substrate level phosphorylation or oxidative phosphorylation, shown here in the cartoon. So imagine that this here, the substrate level phosphorylation shown here, is glycolysis, the most familiar substrate level phosphorylation pathway that you're familiar with, where you start off with glucose, goes to glucose 6-phosphate. So at some point, phosphate is added, and then ATP, the currency of the cell, is generated because that phosphate group is coming off of the substrate. That's why we call it substrate level phosphorylation. ATP is generated from ADP at the expense of a phosphate group within the substrate. Glycolysis, and also the TCA cycle to some extent. Much more efficient is oxidative phosphorylation, where you have, here's a bacterium, a hypothetical bacterium, with that proton motive force. So we've already talked about proton motive force at 2 different points. We talked about proton motive force as a means to drive the flagellar rotation. So protons on the outside of the cell can come across to the inside of the cell to generate torque, to generate rotation in the flagellum. We've also talked about the proton motive force coming across that membrane to bring in molecules like lactose. We talked about symporters and not uniporters, but antiporters. So bacteria, by this pathway shown here, which we'll talk about, which is ATP synthase, can utilize this, in essence, battery, this proton motive force charge differential across the membrane to generate ATP. But now you see that the phosphate group is not coming from the substrate. It's being added to ADP in solution to generate ATP. Much more efficient. And you see that now what's represented on the bottom is a slightly discharged battery. So you have less positive charge on the other side. So some of the potential energy of this energized membrane has been expended and captured in generation of ATP. Now, we're going to talk in this section about metabolism in general, but in reality, what we're mostly talking about is catabolism. We don't have time in this class to talk about the other half of metabolism, which is anabolism. So metabolism is breaking things down to generate energy, to do work. That's the more classic part of what you think about as metabolism. But remember that metabolism also includes anabolism, which, think of anabolic steroids, builds things up. Part of the reason why we spend as much time as we did talking about the biosynthesis of peptidoglycan inside of the cell is that's my one shot at teaching anabolism in this class. It just kind of fit into that section. For the most part, we're going to ignore anabolism from this point. But this metabolism can be broken down into multiple stages. So there's 3 stages of catabolism for energy sources. So in bacteria, this stage 1 is largely done outside of the cells. Remember that part of the reason why we talk so much about protein secretion is that bacteria cannot do phagocytosis. They cannot import large molecules. So instead what they have to do is they have to export enzymes which break down things like proteins, polysaccharides, and lipids, which they cannot transport. But if they break them down into amino acids, monosaccharides, glycerol, and fatty acids, those they can transport by mechanisms that we just talked about, like the ABC transporter pathways, or group translocation and things like that. Then at stage 2, you get, at stage 2, simpler molecules are created to feed into the glycolytic pathway. Not much energy yield in here. Most of the energy yield is going to occur in stage 3, where you have the complete oxidation of these molecules. So you have the glycolytic pathway generating a small amount of ATP. The end product of that acetyl-CoA goes into the famous TCA cycle, which is going to generate all of the oxidations, which are going to generate electrons to feed into the electron transport chain. Here's oxygen as the terminal electron separate generating ATP. This is where most of the energy is generated. And now, many bacteria can do this system very similar to what your cells and my cells are doing. Remember, bacteria invented this. We co-opted it from them. And this is going on in our cells at the because we've domesticated bacteria as our mitochondria. But many bacteria that exist today don't have a TCA cycle. They don't have an electron transport chain. They exist solely by doing fermentation, but they've become really, really good at doing fermentation. So they can survive quite well without being able to do the TCA cycle and cytochromes. The And when we come back on Friday, wasn't even intending to get this far. We'll continue on with metabolism. Remember that next Wednesday, wherever I end on Wednesday's lecture, is the end of the exam material for the following week. So if I go even faster than normal next Wednesday, you know why. Hello, Matthew.